Hello, everyone. My name is Bill Curtis. There is a link to my story below if you are looking for an encouraging example of a person with Parkinson's. The series you are watching is mostly about science. To me, science is about how things work and how can you fix something if you don't know how it works. I plan to make several videos that explain how mitochondrial quality control is key to understanding and treating PD. Chapter 1. Introduction. I am going to describe a way of thinking about Parkinson's disease that focuses attention on how neurons solve the problem of maintaining a healthy population of mitochondria. I am going to explain the jargon superoxide and sentinel. Superoxide is an oxygen molecule, O2, that has picked up one electron, so it has a negative charge. It is also a free radical, meaning it has one unpaired electron. This unpaired electron is inherited from oxygen, which has two unpaired electrons. One of oxygen's unpaired electrons gets paired by the electron that is added to make superoxide. These unpaired electrons will become very important later in this story. So, why did I call it the superoxide sentinel hypothesis? The term sentinel is used in the military as in sentinel duty. A sentinel watches and remains at his post. When a sentinel spots something, he notifies other personnel. We will see that superoxide starts out by sending a message with hydrogen peroxide. Superoxide also awaits a response from the hydrogen peroxide messenger, which comes in the form of a gas, nitric oxide. The term sentinel is used in computer programming. To a programmer, a sentinel is a variable used to stop a process that is repeating. When it is time to stop doing something, the variable is set to the stop value. The code that ran the process is garbage collected to make space for other code. So, if superoxide is present, it is time for the mitochondria to stop working and be garbage collected to make room for new mitochondria. The term sentinel is used in medicine. A sentinel lymph node acts as a detector. It detects the spread of cancer to the closest lymph node. Just as a lymph node indicates a tumor upstream, superoxide is the first reactive oxygen species downstream from damage to the mitochondrion. Now that you know what the jargon in the theory's name means, I will introduce my theory of superoxide sentinel hypothesis of mitochondrial quality control, or SSH of MQC. What are mitochondria? Mitochondria are tiny organelles found inside most cells. They are responsible for the portion of respiration that uses oxygen to make ATP by transferring a phosphate group to ADP. This process is known as oxidative phosphorylation. Outside the mitochondrion in the cytoplasm, glycolysis makes ATP and also provides two molecules of pyruvate to the mitochondrion. Why is it important to remove inefficient mitochondria from neurons? Nerve cells and heart cells have large populations of mitochondria. A heart muscle cell has on the order of 5,000 mitochondria. More than half the cells in our body are red blood cells. Erythrocytes have no mitochondria when mature. A fast-twitch skeletal muscle fiber, used in sprinting, has very few mitochondria. A slow-twitch skeletal muscle fiber, used for posture, has larger mitochondria and many more mitochondria than a fast-twitch fiber. Nerve cells have the most mitochondria of any cell type. A nerve that produces dopamine has hundreds of thousands of mitochondria. Our brain is 2% of our body weight but uses 20% of our energy. The health of cardiomyocytes and dopaminergic neurons is dependent upon the majority of these mitochondria functioning well. This in turn requires that inefficient, low-quality mitochondria are identified and removed. New mitochondria must be made to replace those that were removed. Until now, it was not known how this selection process for removal is achieved. Let's look at the steps. Step 1. Superoxide sentinel accumulates and persists in inefficient mitochondria. Step 2. Some superoxide is converted to hydrogen peroxide messenger and dispatched outside of the mitochondrion. Step 3. DJ1, a sensor of redox potential, counts hydrogen peroxide messengers and available resources, NADPH. If DJ1 finds both, DJ1 becomes active. Step 4. Active chaperone, DJ1, interacts with proteins NRF2, KEAP1, and NF-kappa-B, which turn on genes to make needed proteins. Step 5. Nitric oxide is sent to pass over the mitochondria. The regulation of inducible nitric oxide synthase is complex. In addition to NF-kappa-B, inducible nitric oxide synthase can be induced by bacterial or viral products, cytokines, cell-to-cell contact. Nitric oxide initiator can come from the same cell or a nearby microglia. Timing may be under control of many factors. Step 6. The initiator, nitric oxide, reacts with the sentinel, superoxide. Step 7. The reaction products cause the depletion of a key resource, NADPH, opening an escape valve lowering the proton gradient. Step 8. Pre-station responders, Pink1 and Parkin, sense the loss of proton gradient and assemble the docking station for the phagophore. Step 9. The phagophore engulfs the mitochondrion. Step 10. The activated Pink1 and Parkin also stop holding back biogenesis. So, can the theory be used to explain more about PD? I think the answer is yes. In the context of SSH of MQC, a majority of Parkinson's risk genes can now be understood as playing a role in mitochondrial quality control. These include GBA, LRRK2, PRKN, SNCA, Pink1, Park7, and VPS35, as well as many lesser-known genes. The mechanism of initiation by toxins can be understood through their interference with the superoxide sentinel hypothesis of mitochondrial quality control, or SSH of MQC. The mechanism of initiation by injuries and infections can be understood through their damage to mitochondria. These injuries and infections change the quality of the population of mitochondria. The mechanism behind the Brock staging hypothesis can be clarified. The points of initiation are regions of the brain that are susceptible to loss of superoxide sentinel hypothesis of mitochondrial quality control, or SSH of MQC. This susceptibility has to do with exposure to toxins and pathogens via the gut or nasal cavity. The regions of the brain where PD first shows an accumulation of Lewy bodies have large populations of mitochondria, often with little myelination and high energy demands, making them susceptible to loss of MQC. Progression may be explained partly as a vicious cycle. Once a population of mitochondria reaches a threshold of accumulated inefficient mitochondria, SSH of MQC becomes less effective, causing a downward spiral. Understanding SSH of MQC may make it possible to improve the quality of the population of mitochondria in the brain and partly reverse or slow the progression of PD. If you find this video useful, please hit like and subscribe. If you want to continue following this story, check out Chapter 2 of the SSH of MQC, how the idea for the superoxide sentinel hypothesis came about and some of the people who contributed to the theory.